Hello, everyone, and welcome to this Integrated DNA Technologies webinar on CRISPR genome editing with a high-fidelity Cas9 enzyme. My name is Dr. Hans Packer, and I'll be serving as the moderator for today's presentation. The presentation today will be given by Dr. Christopher Vikalskis, and Dr. Vikalskis is a staff scientist here in the molecular genetics group at IDT, where he works on new enzyme development and purification methods, including the work that led to the Alter SP Hi-Fi Cas9 nuclease. Prior to joining IDT, Dr. Vikalskis was he earned his PhD at the University of Iowa, where he studied genetic regulatory circuits in pathogenic bacterial species. And then he went from there to being an NIH postdoctoral fellow at the University of Florida, where he studied RNA binding proteins and transcriptional gene regulation. Dr. Vikalskis' presentation today should last about 35 minutes, and following that presentation, we will answer as many questions from attendees as possible. And you can ask your questions anytime during or after the presentation. The best way to do that is to type them into the questions box, which you'll find in the GoToWebinar control panel, and that's at the right-hand side of your screen. And if you see there's a little up arrow symbol that you can click that, and that'll pop that window out and make it larger. It makes it easier to type your question into there. So yeah, that's how you'll communicate with us during the presentation. And then at the end of the presentation, I will read as many of those as possible to Dr. Vikalskis, and we'll have a conversation about the topics that you're interested in. Also, in case you need to leave early today or you want to revisit this webinar later, share it with someone, we post all of our webinars on our Vimeo and YouTube channels. We also have a video library on our website, idtdna.com, and if you look under support and education stuff, you'll find our video library as well. So these are the links to our video libraries. We'll be sharing the link to this webinar also by email with you after the presentation in a couple of days. And then we also get asked about slides. We post the slide decks for our webinars at our SlideShare site shown on the screen. And we will actually have that uploaded today, so you'll get a link for that today. Someone is working on that right now. And at any of these resources, you can go look and you'll see we have plenty of CRISPR webinars on related topics, like kind of, how to use our system, how CRISPR genome editing works. So there's a lot of great content already there, plus NGS, qPCR, genotyping. We do a lot of these. So I think that's it for my housekeeping stuff. And with that, I'm going to change the presenter over to Dr. Vikalskis so he can get started.
All right. Thanks for that nice introduction, Hans. And so I'm Chris Vikalskis. I'm a staff scientist at IDT. I'm going to talk to you today about a project we've been working on for the better part of 2 years, trying to develop a Cas9 mutant that reduces off-target effects. And just to give you an outline of what I'll be speaking about today, I'm going to go through some of the basic background on CRISPR, talk about the products that IDT sells, how it works, and then move on to what I think is going to be sort of the main topic today, and that is the notion of off-target editing. And really, what I'm going to hit on the most is that the delivery mechanism matters. And by that, I mean how you're getting the Cas9 protein into the cells, whether you're producing it from a plasmid, from an mRNA, or producing the protein, purifying it and adding it directly, and also how you're getting the guide RNAs into the cell, whether you're producing them off a plasmid, lentiviral, or synthesizing them and adding them directly. So delivery mechanism matters. And then I'm going to move on to talk about a couple high-fidelity Cas9 mutants that have produced and reported in the literature. These were mutants that were made for plasmid delivery of both Cas9 and the guide RNAs, and I'm going to show you some data that says that they don't work as well with RNP delivery, or ribonuclear protein. And the general trend seems to be a reduction in off-target editing at the expense of on-target potency. And then finally, I'll go on to talk about the rationale behind why we decided to make our own mutant, the bacterial screening strategy with which we developed it, and finally go into the data describing how well it works on target compared to the wild type Cas9, as well as these other literature Cas9 proteins by RNP delivery. And then, of course, a discussion of how well it works to reduce the off-target effects, and then go on to some real-world applications of this protein. So we've been sending this to beta testers for quite a while now, as well as doing a lot of in-house testing. So I'll show some editing experiments in primary cells, give you a hint of what it can do in other species, and finally go on to what I think is ultimately what a lot of people are going to do with this, and that is facilitate homology-directed repair. So just to get into the basics of CRISPR, at IDT we sell two different, excuse me, Altar CRISPR products. The Cas9 from Streptococcus pyogenes, as well as the CPF1 protein from Acetaminnococcus species. Cas9, of course, recognizes the canonical NGG PAM. So anywhere in the genome where you've got a di-G nucleotide, you could theoretically have a Cas9 cleavage site. CPF1 recognizes the triple TV or triple T followed by anything other than another T PAM site. Cas9 can leave blunt overhangs, whereas CPF1 leaves 5' overhangs. And essentially all you're doing is using these RNA-guided DNA endonucleases to make double-stranded breaks in live cells. And towards this end, you can either do it to disrupt a gene's function by allowing the native non-homologous end joining pathway to repair, ultimately resulting in some sort of small insertion or deletion, otherwise known as an indel. And alternatively, you can replace whatever it is that you're cleaving with homology-directed repair, as long as you include a donor DNA, whether that's in the form of a single-stranded oligo or a double-stranded fragment. So we're selling the Altar system, and this is a two-part RNA system for Cas9. So Cas9 will be the majority of what I talk about today. And in this two-part system, this closely reflects what actually happens in the bacterial cells in the primitive immune system, where you've got a targeting CRISPR RNA or CRRNA. In this case, we are selling a 36-mer with 20 nucleotides of target sequence and 16 nucleotides that hybridize to the universal tracer. And this 67-mer universal tracer RNA, of course, hybridizes back to the targeting RNA and also interacts with the Cas9 protein. And it's also true that people have appended these two RNAs to form a single guide RNA. And I'm going to show you some of the benefits and disadvantages of either system. So at the bottom here on the left-hand side, you can see the two-part system where you've got this 36-mer targeting cRNA hybridized to the universal tracer. And then the single guide form where you've just got this sort of loop here in the full RNA molecule. So for the two-part system, these are not ideal to express in the cells from plasmid DNA. You would need to add on transcription terminators, things like that. For the same reason, not ideal for in vitro transcription or I guess IVTs. We're an oligo manufacturer. They're very efficient to make with chemical synthesis, and we've been able to truncate them and make them a good deal shorter and still maintain or even in some cases increase potency. So for the cRNA, we've got this 36-mer, and since it's so short, we can make it on the cheap. The 67-mer tracer, we can make it cheaply as well. And because we're making them in a chemical fashion, we can modify chemically any position we want to do things like allow for escape of the immune response. Whereas with these single guide RNAs, these are ideal for DNA expression cassettes. You can put a terminator on, they're going to be made in vivo. For the same reason, they're ideal to be made from an in vitro transcription reaction. That's relatively low cost. We find they're inefficient for chemical synthesis, mostly due to the length, they're longer, and then also the higher cost. And then if you're making them chemically, it's costly to do this. And if you're making them with in vitro transcription reaction, it's very, very difficult to modify, especially the internal nucleotides. So for that reason, we do favor the two-part system, and we also favor it because we see a reduction in toxicity and induction of the innate immune response. So this is just an example, an experiment here where we're showing you a HEC293 immortalized stable cell line that constitutively expresses Cas9. You can see the cells look quite healthy. If we take an sgRNA that's produced from an in vitro transcription reaction, you can see a marked amount of cell death. And if we do the same experiment but use the two-part system, you can see the cells are mostly indistinguishable from the negative control. So we also see evidence for induction of the immune response. These are qPCR reactions, and we're looking at one specific marker for the induction of the immune system. And on the left-hand side here, you're seeing a human normalizer gene. So we expect, and in fact, we see everything is sort of equal. This is the IVT sgRNAs, the two-part and the cells only are all right on top of one another. And then for the actual immune system marker, you're seeing the two-part system and the cells only negative control are on top of one another, whereas the sgRNA IVTs, you see this huge CQ shift indicating a large induction of the immune response. And this is also true, we've tested this with a lot of other immune system markers as well, and we just don't see this with the two-part system. Nevertheless, you can implement CRISPR-Cas9 cleavage in a variety of different ways. You can supply the Cas9 protein either as a purified component, adding it directly to the guide RNA complex, and then transfecting the resulting RNP. You can supply Cas9 as part of the stable cell line with the Cas9 gene integrated on the genome. You can use in vitro transcription to produce a Cas9 mRNA. And alternatively, you can use either a plasmid or a lentiviral delivery system to supply Cas9. And it's basically the same story with the guide RNA complex. You can use our ALTAR two-part system. You can use the aforementioned sgRNAs. You can also produce an sgRNA by transfecting a double-stranded DNA fragment that encodes them, or alternatively, as for the Cas9 protein, you can use a plasmid or lentivirus. And you can sort of mix and match these however you want. And we, of course, favor the two-part ALTAR system, recommend it with RNP delivery, and really it couldn't be simpler. And in fact, all you do is order your targeting CRRNA, mix it into 1-to-1 molar ratio with the universal tracer. You hybridize it by heating to 95 degrees for 5 minutes and allowing slow cooling on the benchtop. In a few minutes after cooling, you can add the guide RNA complex from this reaction to purified Cas9 protein, again at a 1-to-1 ratio. Let that form an RNP for 10 minutes, and then the resulting complex can be delivered by whatever your favorite delivery mechanism is. So we like this system. It's very, very easy to do. You escape the immune system. You don't have as much toxicity, but there's also another reason we like it. And this all hovers around the idea that the delivery mechanism matters, and most prominently for the reduction of off-target effects. So Cas9 from Pyogenes is known to produce these off-target effects. And what I've got on the screen here is an example of the GuideSeq work from Keith Young's lab at Mass General. And what they show is that if they include in their CRISPR-Cas9 editing experiments, along with this EMX1 guide, a double-stranded DNA fragment, and allow that to integrate at all the double-stranded breaks with non-homologous end joining, they're able to sequence and find all of the double-stranded DNA breaks that were in the cell. And they see, of course, that this on-target site, the intended edited site for EMX1, is the most frequently edited site, but they also find all of these other off-target sites, some of which have a relatively high frequency of editing. And you can see, as expected, these are very similar to the intended on-target site with a few changes. And what we've sort of found in-house is the extent of off-target editing seems to correlate with the delivery mechanism of both the guide RNA complex as well as Cas9. And it seems to be worse when you've got plasmid-expressed guide RNA and Cas9. And I think I could, go on and try to advertise for the system we sell, but I think ultimately people are going to believe this if they see it not from just our group, but from our friends at Thermo Fisher. And what they've shown in this very nice experiment is a Western blot where they show you where Cas9 is and how long it persists as a function of time and also as a function of delivery mechanism. And they correlate that with detecting levels of off-target editing. And what they show here is with DNA plasmid-based Cas9 expression, it actually takes a few hours to get Cas9 present to its steady state level in the cell. And then it's also stable outwards to 72 hours. So it comes on pretty strong after some time and is not easily gotten rid of. With mRNA, you seem to see Cas9 produced a little sooner, but it also seems to be very stable as a function of time. And then if you purify Cas9, form a guide RNA complex in a test tube and transfect that directly, you can see that you get a lot of Cas9 protein that's present very early, and that is either degraded or diluted by growth over time and is almost gone by 72 hours. And what I think is so cool about this is if you correlate that with off-target editing, you can see that in black here with the plasmid DNA, these two known off-target sites, you get the most off-target editing with plasmid delivery when Cas9 is most abundant and hangs around for a long time. It's sort of an intermediate effect when you're looking at mRNA, which sort of makes sense. And then if you look at protein, where Cas9 does not hang around as long, you see the lowest levels of off-target editing. And we essentially see the same thing when we do these experiments here at IDT. And what I'm comparing here is low-level constant expression of Cas9 in this HEC293 stable cell line. And I'm comparing that to RNP or ribonuclear protein delivery of Cas9 in the same EMX1 guide. So what you're looking at here is in dark blue is the intended on-target site, indel formation at that site. In light blue is the top known off-target site for this guide, and in gray is the second known off-target site for this guide. And simply switching the delivery mechanism from stable cell line to RNP, you maintain the same level of on-target editing, but even at the highest dose, you dramatically reduce off-target editing. And then what's really interesting about this is you can dilute that complex even further, for the most part, maintain on-target editing until you get to this lowest concentration, but you can also reduce off-target editing even further. So this all sort of hits back on the same notion that the delivery mechanism does indeed matter. And if reducing off-target edits is your goal, using RNP can get you a part of the way there. And as you might imagine, in a perfect world, we'd be able to sort of predict what the best guide is going to be, which ones are going to have the fewest known off-target effects, and choose those guides and not have to worry about any of this stuff. And in fact, there have been several very good attempts at making prediction algorithms to determine whether guides are good or bad. And what we found is that it's just very, very difficult to predict, accurately predict Cas9 off-target sites. And I think this has a lot to do with there just being a lot of things that we don't yet understand about cleavage preferences for the Cas9 enzyme. And I think sort of that basic concept is best shown in this experiment. So we've taken a cell-free Cas9 cleavage experiment here. This is very similar to digenome-seq or site-seq. It's a proprietary system we have in-house. We're using this androgen receptor guide. And if you look at the guide sequence, there's nothing obvious about this that would say, it doesn't seem to be horribly repetitive. There's nothing obvious about it that says this is going to be a bad guide. And what we've done here is we've shown all the cleavage products from this cell-free experiment and put them in rank order. I've colored the on-target cleavage in red here, and thankfully that is the most frequently edited site. But you can see all these different off-target sites in blue and green. And what is the most scary thing about this diagram is that I've colored in green the ones that are predicted by the SVM algorithms. And it's less than 10% of the total off-target editing. And I think that this is certainly a worst case scenario for off-target editing in that, you know, there's no transcription factors, there's no chromatin around since this is purified DNA. So, you know, Cas9 is more free to do editing. So again, this is a worst case scenario. And also it's going to hang around forever. You're not going to get degradation of the RNP complex. It's not going to get dilution by growth. But even considering that, these most frequently edited off-target sites were not predicted by the off-target algorithm. So I think, you know, if you're in a position where off-target editing is a concern, this is kind of a scary graph. And there have been a number of attempts to reduce off-target effects. And obviously we found that delivery of Cas9 and the guide RNAs as an RNP really gets you part of the way, but it's not a total solution. It doesn't eliminate off-target editing. And we've also tested in-house some of these other mechanisms to reduce off-target editing, like reducing the length of the targeting CRRNA. So normally we use a 20-mer, and there have been some literature reports of shortening that to 19, 18, or even 17 nucleotides. And we find that that has, you know, it's sort of a mixed bag. We find that for some guides, it does indeed reduce off-target editing and you still get good on-target editing. But it's really sort of unpredictable. And a lot of the guides that we tested, you see a marked reduction in on-target editing, and that sort of renders it, not a good option. And I think it's sort of the same thing for chemical modifications. So we've tried to introduce specific chemical modifications at different points within the targeting CRRNA, and it's, sometimes we see a reduction in off-target editing, but oftentimes that correlates with a reduction in on-target editing. And so that leads us to the sort of last thing on my list, and that is the so-called high-fidelity Cas9 mutants. And at the time we started doing this work, throughout most of the development, there were only 2 of them. There's now actually a 3rd. But the 2 proteins that were engineered were the Cas9 protein from Feng Zhang's lab at the Broad Institute, and then this Cas9HF1 from Keith Young's lab. And these were a really fantastic and clever way to address this problem, where they looked at the crystal structure of Cas9 in complex with the RNA and DNA target site and made pointed mutations to reduce the affinity for either the targeting or non-targeting strand of DNA. And I think the idea was to eliminate as many of the off-target weaker interactions as possible and still preserve on-target effects. And in the papers, these were all done with plasmid delivery of Cas9 in the guides, and they seem to work very, very well in both cases. And before we got into the business of trying to make our own proteins, we thought that we would test both of these and find out if they worked as well with RNP delivery, because that's our favorite way to do things. So we introduced the amino acids that comprise these particular mutants in the context of our Altar SP-Cas9 protein, purified them to homogeneity by the same process as the commercial material we sell. And then we tested them to start off with to look at on-target performance, comparing, and I think we've looked at greater than 50 cRNAs and greater than 10 genomic loci at this point. And in the experiments I'm going to show you, we've tested these comprehensively, looking at immortalized cells with liposuction, and then also T7E1 is sort of the readout for this. But keep in mind, we've also repeated a lot of these experiments with both electroporation and also using NGS as a readout. And what we found from these experiments is actually quite striking, and that is, so on top here is our 2-part Altar system. Everything delivered is a 10 nanomolar RNP by liposuction. These are 12 guides within the HBRT locus, and again, these are in immortalized cell lines. What we find is that the wild type protein, for the most part, seems to function pretty well with the majority of these guides, save for a few. And the ESP-Cas9 protein seems to work only at about 50% of sites, taking a massive hit at some of these latter HPRT sites and a few in here. And the SP-Cas9HF1, again, when delivered as RNP, seems to only work at a fraction of these sites. And again, I don't think that's a failing of these proteins so much as just a statement of how they were made. They were made for plasmid delivery of Cas9, and that is indeed how they work. And just to show that, this is sort of a universal thing and not limited to the product that we sell, our good collaborators at Stanford, Danny Dever and Matt Porteous, also put these into human primary cells instead of immortalized cells. And they used chemically modified sgRNAs instead of the 2-part system, and also electroporation and analysis by NGS. And they show basically the same story. For the majority of these clinically relevant loci, the wild type protein works pretty well, and there are some significant hits in on-target editing with both of these proteins. And at the time, we really wanted to pick the best of these and hopefully license and make a commercial product. And what we found is that they didn't work well as RNP, and in hindsight, this makes sense. They were made for plasmid, where you get continued and long-lasting Cas9 synthesis. And we didn't want to use plasmids because they're prone to toxicity and immune system stimulation. So none of them worked well. Reduced off-target editing occurred at the expense of on-target function. And my background is that of a bacterial geneticist. I've done a lot of bacterial screens, transposon hunts, directed evolution, and I thought that, okay, maybe I can solve this problem by doubly selecting for mutants that avoided off-target editing, but also were able to maintain on-target potency. And the bacterial system we used is simply a 2 plasmid system here that I've got pictured in this cartoon. And in the first plasmid, you have an on-target site that you must cleave to avoid the expression of a bacterial toxin that's present. So you have to cleave this plasmid if you are to survive. And then on the second plasmid, you have an off-target site, a known problematic off-target site for this guide, and an essential gene that the organism needs to survive. So you have to avoid cleaving this plasmid and successfully cleave this plasmid. So we made a library of Cas9 mutants with low fidelity PCR. We screened about 250,000 of them. We got roughly 100 hits from the screen, and we put it sort of through the ringer, changing on and off target site pairs, doing repeats, et cetera, and ultimately purifying and testing the best of these. And this is sort of the data that came out of that original primary screen. And this is, again, rank ordered by what I'm calling a discrimination ratio. So that's the ratio of on to off target editing for this particular guide sequence. So the higher the number, the better the particular mutant discriminates on from off target. And these sites were picked because wild type does not actually do a good job at discriminating at this particular guide. And you can see out of this screen, we were able to select a handful of these that actually had a phenomenal increase in this discrimination ratio, and several others that were sort of middling effects. But we tested the best of these, purified a handful of them, tried different combinations, and we ultimately settled on one particular attractive mutant, and we tested it using the same sort of experiments that we examined the original high fidelity proteins with. And we did this again, liposuction at the HPRT locus with the RNAi max reagent from Thermo Fisher, immortalized cell lines, and also our good collaborators tested with electroporation into, well, that's not right, into primary cells. And what we found is really striking, and that is that our Altar Hi-Fi protein, our best mutant here in orange, functioned the same as wild type at the majority of these HPRT sites, save for a handful. And if we look at the same experiment, but delivered with chemically modified sgRNAs, delivered with electroporation into primary cells by our collaborators, we see it's basically the same story. Our protein is largely indistinguishable from the wild type. And if you're going to, show that you've made this protein that reduces off-target effects, I think you have to actually show that it reduces off-target effects. So what I've got here in this experiment is sort of a crude analysis of how well this protein functions to reduce off-targets. And these are 3 guides with 3 on-off target pairs that have previously been reported in the literature and either the GuideSeq work or the other high-fidelity protein papers. And what we're showing is that this EMX1 locus, you see that all 4 proteins, wild type, the 2 literature proteins, and our protein all function pretty well at the on-target site. For the problematic known off-target site, you can see even wild type by RNP delivery does not produce a lot of off-target editing. With this assay, we see no detectable off-target editing with all 3 high-fidelity proteins. And then interestingly, if we move over to this hex site 4 guide, we found in-house that the wild type protein produces a lot of off-target editing, and this is actually a challenging off-target site to beat. It's interesting that we show that, again, the literature proteins don't do as well at this site for on-target with RNP delivery, whereas our protein does quite well. And then all 3 with this assay show no detectable off-target editing. And it's basically the same story with this VEGF-A3 guide. Ours looks virtually indistinguishable from wild type. The other high fidelity mutants that were developed by plasmids don't work as well with RNP. And then all 3 proteins reduce off-target editing below the limit of detection of this assay. So this is a very blunt instrument for looking at off-target editing. Ideally, you would want to do it in an unbiased genome-wide fashion. So we're going to go back to this cell-free Cas9 cleavage system that we've made in-house, and again, this should be an overestimation of the amount of off-target editing with this protein. So we think that this is really putting it through the ringer. You're not going to have degradation of the RNP because you don't have proteases and nucleases. The genomic DNA is going to be absent factors that would encumber Cas9's binding. You don't have chromatin, you don't have transcription factors. So we tested 5 unique cRNAs that target distinct genomic loci, and I'm going to show you a summary of the data, editing data from these experiments, and then hone in and show more detail on one in particular. So what you're looking at here are pie charts where the blue represents the fractional percentage of on-target editing at these guides. So with the wild-type Cas9 protein, you can see we get about 15% of the total reads are on target, whereas you get about 85% here in orange are the total sum of off-target editing events with this particular androgen receptor guide. And this is actually one of the first ones we looked at in this fashion, and with relationship to these other experiments, it was almost kind of shocking to see that it only brought on-target editing up to 52%, but I'll show you more detail about why this is in just a moment. But looking on some of these other guides, the EMX1 guide only edits on target 15% of the time. Our Hi-Fi protein brings that up to almost 100%. The beta-catenin guide we're using here edits 19% of the time on target. Ours brings it up to 90%. This GRHPR guide is actually pretty good from the start. It edits on target 72% of the time. Our protein brings it up to 100. And finally, this MET guide is around 29% editing on target, and our protein brings it up to 90%. What I like about this series of experiments is I think if we're anything at IET, we like to be honest arbiters of the facts. We want to show things for what they really are. And in this experiment, you see some examples where you've got a really poor guide, and I'll give you more detail on this specific one right away, where our protein does an admirable job, but it's not perfect. And you've also got some guides like this GRHPR guide that are great from the start, but our protein just gives it a little boost. So let's look a little more closely at this androgen receptor guide. And this is the same sort of plot I showed you before, where we've put the total cell-free editing sites as a function of rank order. And I'm showing you data from the wild type protein in blue and our high-fidelity Cas9 in orange. And just to point out again, I don't think that there's anything from this particular target site sequence that screams that this should be a bad guide. And what we find is that if we've got the asterisk on the on-target site, there's not much difference between the two proteins for on-target, but you can see all these off-target sites in blue, and the vast majority of which are eliminated by using our high-fi Cas9 protein. So even in one of the worst case scenarios where you've got a really, really bad guide, most of which these sites probably don't actually happen in the cell, our protein does quite well. And we thought that this was really putting the protein through the ringer, but ultimately, we should look at this in cells. So the best way to do that so far is with the GuideSeq approach. And what we found with GuideSeq is that it does a fantastic job at identifying what the off-target sites are for a given guide. But where we struggle to use it in-house is by giving quantitation to these numbers. So we don't find that these numbers are very quantitative when we try to assess them with PCR amplification-based techniques. So what we've gone on to do is to use GuideSeq to identify what these sites are, and then we use a proprietary multiplex PCR coupled to NGS sequencing approach to actually quantitate the frequency of editing at each one of these different sites. And so far, we've done this in-house with the EMX1 guide that's been done in the literature, and I'll show that data today, and then eventually we're going to publish this information, and we'll have a variety of different guides tested with this sort of comprehensive analysis. And what's so interesting about this is so remember, these experiments from Keith Young's GuideSeq work were done with plasma delivery of the guide and Cas9. And when we did this in-house, we did it with RNP delivery, where we know this eliminates a lot of off-target editing. And what we found is that we were only able to detect editing at the on-target site and these top two off-target sites when we use RNP delivery and the GuideSeq method. And this is the data from that set of experiments. And we did this with either the wild type Altar SP-Cas9 protein or the new Hi-Fi protein. And just to point out, this is a log scale graph here. And what we see is that if you take with RNP delivery using this very quantitative assay and look at on target in orange, the number one off target in this light blue, and then the second most frequently edited off target site in gray, you can see that simply diluting the RNP complex here gives you quite a reduction in off-target editing and not a huge effect on on-target editing. This is basically what I showed before. But now if you switch to our Hi-Fi Cas9 protein, you only see a whisper of editing at this most frequently edited site, and it's vastly reduced at this high concentration. And simply diluting the complex a little more, you don't suffer for on-target editing, but you eliminate detectable off-target editing. So this is very, very exciting to us. We were very happy with these results. And we actually had been sending this protein to beta testers so that we have data not just in our hands. And some of the early applications of CRISPR-Cas9 are going to be these ex vivo genome editing therapies. And in particular, our close collaborators, Danny Dever and Matt Porteous at Stanford, are looking at editing of the beta globin 1 gene, which is associated with sickle cell disease and beta thalassemia. And what they have here in this figure is in the upper left-hand corner shows you the target site they are trying to edit in this HBB gene. And what they found is that there's a lot of off-target editing which was found by GuideSeq at this particular site. And it is identical to the on-target site, save for three changes in the 5' most end of this target site sequence. And they find that even with RNP delivery into primary cells, there's quite a bit of off-target editing at this site. So looking at the quantitation by NGS, in 3 replicates with the wild-type Cas9 protein, you see greater than 80, almost 90% editing at the intended on-target site. But for this known off-target site, you can see the values range from 30 to 50%. So quite a bit of concerning editing at that site. And as you can imagine, this would be sort of a challenging off-target site to beat since the changes are so far away from the PAM or the seed region of the guide. So we sent them our Hi-Fi Cas9 protein, and what they show is that they largely maintain on-target potency with our protein, and they reduce off-target editing significantly. And over here is a quantitation of that data. You can see with our Hi-Fi protein, they maintain 75 to 80% editing, and they reduce 30 to 50 down to 1 to 3% editing. So they see a really strong reduction in off-target editing. Many of these sites, other sites that were either found in GuideSeq or found via prediction, are sort of functional zeros, but there are a couple examples here of sites that are edited 0.6 to 0.2% of the time that our protein also reduces to a functional 0. And what I think is also very exciting about this is not only did they look at indel formation, but they also looked at HDR, or homology-directed repair in primary cells at this locus, and they see that they maintain approximately the same amount of HDR using our Hi-Fi Cas9 protein. And then they've also gone on, so this is the indel formation through these other clinically relevant guides that I've already shown. They've also gone on to do HDR experiments at all these different clinically relevant loci as well. And they show basically the same story that we've been saying all along, is that our protein maintains a lot of its on-target potency, even with HDR at these different clinical sites. And the other high-fidelity proteins, which were made by for plasma delivery, seem to show a marked reduction in HDR. And we don't have any off-target data yet to look at our Hi-Fi protein in mice. That's something we're working on pretty hard. But we have taken a look at how well it functions on target at several different sites within several different genomic loci. And we're very pleased to find that it seems to work on target very well. We don't expect any surprises for off target since we've already sort of looked at it in a totally cell-free context. And with that, I want to make a few concluding statements, and then I'll end on one experiment that I think sort of pulls everything together. And so off-target effects are a very real concern when using CRISPR. They're potentially a hidden disaster. Simply switching to RNP delivery can get you a good bit of the way there to reducing off-target effects. A lot of the mutations in Cas9 that have been made to reduce off-target editing, depending on the delivery mechanism, these can also hurt on-target cleavage. We've used a bacterial selection scheme to find a protein that maintains the best on-target editing while reducing off-target risk. And I think just to be practical, I mean, there's no way that we're going to ensure 100% on target and completely eliminate off targets, but I think you can get part of the way there by using RNP, carefully selecting a target site, and hopefully we can develop better algorithms in the future. And finally, by using high specificity enzymes like our Altar Hi-Fi. And I think that this experiment sort of pulls everything together. And this is, again, an editing experiment with EMX1. And what you're looking here is, again, at a log scale graph, looking at the on-target site, the top off-target site, as well as the second most frequently edited off-target site. And you're looking in dark blue at the wild-type Cas9 produced in a stable cell line, where you expect off-target editing to be high. You're looking at wild-type Cas9 delivered as a 2 micromolar RNP, and then our Hi-Fi protein. And you can see all three of these proteins and delivery mechanisms give you good on-target editing. You can achieve that anyway. But if you look at off-target editing, you get the highest amount with Cas9 produced in a stable cell line. Simply switching to RNP delivery for either site gets you a good deal of the way towards reducing off-targets. And then finally, switching to our Hi-Fi protein and diluting and doing a careful dose response gets you the rest of the way there. So we're very, very excited about this product. It's been a long time coming, and I'm glad we're finally able to release it onto the market. And I'd like to thank everybody for listening in, and I'd be happy to take some questions.
I guess it's not surprising that you're excited about that since you've done so much work on it. There's that. Hey, so we have plenty of time for questions here, and if you have a question for Dr. Vikalskis, you can ask them right now. You can type them into the questions box, and we will read through as many of them as we possibly can. I also wanted to draw your attention to the chat box really quick. We've posted the slide deck for today's presentation, and you can grab that right now. If you don't want to look at it right now, we will also be sending you the link to it by email, so you don't really need to worry about that. Just it's available to you if you want it. Okay, so here's a question, and this is a great question just in the context of all of this, talking about design algorithms and the need for a Hi-Fi Cas9. It used to be that there was like a very clear idea of the importance of the position of those variation bases. You know, like if there's a variation that's far away from the PAM site, it doesn't, you know, have as much of an effect as if it's close. But is that difference also hard to predict now? So,
I mean, you know, the truth of the matter is I just don't think we have all the data we need to make those conclusions. But what I will say is I suspect it's a combination of multiple things happening at one time. I think the traditional rules apply where, you know, the closer a change is to the intended target site, the closer it is to the PAM or the seed region, the less likely it is to be an off-target site. But I think you probably have other factors like, and this is just speculation, some basic cleavage preferences for the Cas9 enzyme. So these are things that happen when Cas9 already gets there. Maybe it has a preference for a specific nucleotide at a specific position, and under the right circumstances, that can sort of trump simple, you know, binding interaction kinetics. I just think there's a lot of things probably happening, to borrow the phrase, you know, sort of in a mosaic, and it's really hard to decipher, you know, what those rules are and so forth.
Sure. Okay, so I'm not sure how much of this you can talk about, but I'll ask the question anyways, and maybe you can give a little bit of detail here. How many mutations does the Hi-Fi Cas9 have?
Yeah, so so that is something I am not able to talk about today, but what I can say is that we are at heart an academic organization. That's how we were founded, and we do intend to publish this as quickly as possible. And you know, obviously, we're going to identify what the position or positions are at that time.
Okay. Yeah, no, that's a great answer. Actually, it's nice that we are planning a publication for that.
Yeah, absolutely.
This is a question that we do get from time to time. I'm not sure. I haven't heard a recent answer for this, so it's good to be asking it again. Do we have any recommendations for people for applying RNP delivery in plant genome editing?
Yeah, so that is something that our protocols are sort of not quite there yet on, but it is actually, so that did come up at a recent conference, and it's something that we're working very hard to address. And I think that what's interesting is, a lot of people that are working in protoplasts, they're finding that plasmid-based delivery of Cas9, the protein isn't made fast enough for how quickly you have to manipulate the protoplast. You need Cas9 expression, you need the editing to happen quickly. So people tend to apparently favor RNP delivery in these particular protoplasts. So, that's become something that's of high importance to us, and I think we should be able to address it, hopefully in the near future.
Okay. This is interesting. Do we have anything about Cas13? Are you familiar with that?
Yeah, so this is the if I'm not mistaken, the RNA editing technology that's been sort of applied to a diagnostic setting. We don't have a product for that at the moment, but we always have our ears open to new and exciting technologies, and obviously there have been a couple great papers about this technology, and it's something that we're looking at very carefully.
I apparently need to go do some reading. Let's see here. There's a few similar questions. People really want to know about they want to know the details of the enzyme itself, sequence and changes and whatnot. So yeah, look for that paper in the future coming from Dr. Chris Vakulskis. Apparently will be one of the authors on there.
Hope so.
Probably first. I would assume. I would assume.
But really, we want to publish this as quickly as humanly possible. So that information is going to be out there.
Okay, so this must be for the Staph aureus, I'm guessing. Do we have Hi-Fi Staph aureus SA Cas9 doing a similar job?
We haven't. We certainly don't even have a commercially available wild type Staph aureus, and I think if memory serves, that has a more restricted PAM site. So that's not something that we've done a lot of testing with. But I can't say for certain, but that might be the sort of thing where we'd be open to looking into that. I expect the amino acid positions are likely conserved. So yeah, we may be open to that, but right now we don't even have a wild type SA Cas9.
Okay. Have we contemplated working with the Hi-Fi Cas9 to create nuclease variants?
Yeah, that's an interesting idea. We've also thought about the dead Cas9 and introducing a Hi-Fi in that context as well. But our thinking on that so far has been with the nucleases. In doing HDR with these dual guides, we kind of didn't see the need to increase the fidelity. Since you're only nicking, and the only way that you're going to get an actual double-stranded break is if you have two entirely separate guides in close proximity. So I guess what I'm saying is the nuclease-based HDR or cleavage approach is inherently high fidelity, and we haven't so far seen the need to increase its fidelity. But again, we're sort of always open to this stuff. So.
Right. And did you want to say a little bit about the nucleases that we also have? Yeah.
So we at the same time we released the high-fidelity Cas9 enzyme, we also began selling the D10A and H840A nucleases. We actually have some good protocols for those available on the web. You feel free to visit the website and look at the data, and if anybody is looking for them, we have them.
Okay. This is an interesting one. So have we tried plasmid delivery of the Hi-Fi Cas9? And I think you touched on that a bit. And then there's a second part, which is how does it compare to ESP Cas9?
Yeah, so we have tried plasmid delivery, and in fact, when you're doing screening and that sort of thing, it's just easier to do all this stuff on plasmid. But the caveat there is, when we were doing those experiments, we supplied the guide RNAs with our two-part Altar system. So we didn't supply those on a plasmid. So with that approach, our protein actually worked pretty well, even with plasmid delivery. But honestly, we didn't do a real careful comparison of it to the other high fidelity mutants because our main interest was looking at it in the context of RNP delivery. But I can say it's not, it's not like ours just doesn't work with plasmid. I suspect where it would not do as well relative to the other high fidelity proteins is if you were delivering both components by plasmid. And in that case, I think, the other high fidelity proteins are probably a better option anyway, because they were specifically evolved for plasmid. So
I've been doing these webinars for a while, and I'm going to tell you that I have thought a lot more about plant stuff through this process than I would have otherwise. So this is kind of how I get educated about plant stuff. Somebody is asking, and this is probably going to be out of your area of expertise, but is there any reason why off-target activity would be less important for plants that you can think of?
I don't know enough about how easy it is to breed out unwanted off-target edits. And I think, going back to the idea that delivery mechanism matters, it's possible that either the protein, the Cas9 protein, or the guides are less stable in particular plant species. Maybe they're more rapidly diluted out by growth, although that's kind of hard to imagine. I'm sure there are some cell lines for some organisms where the RNP doesn't hang around very long at all. Maybe it's very, very short-lived. And in that context, you would imagine that you would get the lowest amount of off-target editing. But unfortunately, I just don't know enough about plants to intelligently answer that question.
Right. And like I said, I mean, I've certainly become more thoughtful about plant questions because we do have so much interest in that. It's obviously agricultural research is a really important area, but that's not where I'm trained. So
absolutely.
This is a question we can possibly ask to some other people. We happen to have a product manager who has a PhD in plant biology, so that might be a question for him. All right, another question here that I'm not quite sure of. It says, just to clarify, targeting a single base pair mutation for repair in a cell line is not exactly feasible at the moment. And that's a statement with a question mark at the end.
Could you repeat that? I'm not sure I fully understand.
I'm thinking that what this person is asking is, so it says targeting a single base pair mutation for repair within a cell line is not exactly feasible at the moment. And I'm guessing that they're saying that, sure, you can correct that, but you can't really do it precisely at the moment with current technology.
Well, all I can say about that is it would be difficult to select for such an event. Ultimately, what you would have to do is get, an HDR. Well, I can think of two strategies to do it. One is HDR, and since there would not be any selection for a single base change, because obviously you can't incorporate an antibiotic resistance marker or anything, you'd have to have a pretty high frequency HDR event and then be able to isolate the clone by limiting dilution. But the other way is there are also these so-called base editing enzymes that have come on the forefront, where you're taking a D-Cas9, so a dead Cas9 coupled to a cytosine deaminase, and in those particular with those particular recombinant forms of Cas9, you can actually introduce a specific point mutation in the absence of HDR. So, I think it is possible. I just don't think it's easy yet.
Right. That's actually, yeah, that's interesting. Okay, so do we have any recommendation for finding specific guide RNAs in plants without the total genome being sequenced?
Oh boy. I don't know what the best answer to that is, other than trying to empirically determine what the total number of off-target size is. And the existing technologies, obviously the majority of what's done is done in immortalized cell lines. So I don't know how easy it is to do a guide seq in plant cells. I do think that the available algorithms are going to help you identify the most obvious off-target sites. And I think, that has to be a component of every one of these experiments. And I think eventually the algorithms will get better. And I guess my only recommendation would be to try to do it empirically, and something like a guide-seq would allow you to do that without sequencing the entire genome.
Hey, here's a completely interesting question, and it's not exactly science. I haven't thought about this. Do you know, does somebody need licenses to use our enzyme?
I think when you purchase it, you're acquiring, and don't quote me on this, but I believe you're acquiring an individual use license. But I guess it depends on who it is, right? If
Exactly.
you're talking about on a commercial scale, that's probably a different story.
Here's the thing, yeah, if you have this question, it is, I think, going to depend somewhat on, what you plan to do with it. So like if it's another company that plans to use this, it's possible. If you have, if you're trying to use it outside of, like the research use only that that we offer it for, then you're definitely going to want to talk to somebody here. You can find the right person at IDT to talk to by going to our website, idtdna.com/contact us. And, somebody can help you talk to somebody specifically about a legal concern like that. But for the most part, for just research use, if you bought the enzyme from us, you can definitely use it for basic research. We'll definitely follow up with this particular question. I haven't seen that before, but it's obviously a concern to people.
Yeah, absolutely.
Okay, let's see here. This next question is, have you done additional rounds of directed evolution for protein engineering using your surprising finding of androgen receptor off-target activity?
So maybe have we used that guide as something to pan the directed evolution against?
Oh, sure. So they're asking, and that is the rest of the question that they're asking is, can you make an even more high-fidelity Cas9?
Yeah, I suppose it's possible. The trouble we've run into is the more we mess with things, the more we reduce on target activity, and you very quickly lose significant on target activity. And I think we put a lot of effort into making more specific versions that ultimately we ruled out because they just lost too much on target activity. And
you're looking for the best balance.
Yeah, the best balance, and I think that's what we found here. And it's not something that we're ruling off the table. And in fact, that's a good idea. That androgen receptor guide would be a good one to look at.
Okay. This is a question. This is a good question too. What is the impact of the cell doubling time for the RNP efficiency, and is it still working with cells that have a week of doubling time?
Man, that's just something I don't think we've looked at. That's a good question. We haven't looked at that carefully. I can say that RNP delivery, the cells are very, it's more finicky. Especially with liposuction. So you need to take cells that are not fully confluent yet and that sort of thing. But we really haven't done a careful analysis of cell doubling time and editing efficiency.
We generally look at the results of our editing, though, at 48 hours, right?
Mm-hmm.
So, maybe there's a partial answer in there. Like for us, we get our editing results within 48 hours. Maybe that's a helpful way to kind of gauge that.
Yeah, and I think we need to go out to 48 hours to see the highest levels of editing. But also I kind of wonder if that's going to depend a lot on the cell line and species as well. So
okay. Okay, here's a question for you. And it says, what is the recommended guide length when using the Hi-Fi Cas9?
So for the Altar system, we're recommending 20 and no shorter.
And so that's the guide length, but also, we really do recommend, using the two-part guide RNAs that we offer, just because, I mean, they've also been optimized for other things, including they're length optimized, they have the modifications and whatnot to prevent that immune response and such. So that's why we really do recommend those.
Right, so yeah, so 20 nucleotides of guide sequence give you the absolute best results.
Okay, so, all right, we've reached the top of the hour. There are still a few questions that have not been answered, and unfortunately we can't fit them all in, but we definitely will follow up with everybody by email. If you feel like you would like to talk to someone, you can go to that idtdna.com/contact us, all one word, no spaces, no other characters in there, and just, yeah, reach out. Otherwise, yeah, we'll be emailing you and you should get an answer to your question. You'll also be getting a follow-up email with links to the presentation, and the slide deck is already available, but you'll get links to the slide deck as well. And really, thanks everybody. They're really great questions for this Q&A session. And Chris, it was an excellent presentation, so thanks to you as well.
All right, thanks everybody.